Hello, everyone. Welcome. And if you celebrate, happy St. Patrick's Day. I'm Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to this webinar. It's presented by ASCCT and the European Society for Toxicology in Vitro. I encourage you to visit our website, which you can see on your screen there, to find out more about our organizations and consider becoming a member to support programs like this webinar series. Both of our organizations host regular meetings and pursue other activities to promote the fields of computational and in vitro toxicology and aim to support early career and all scientists in their endeavors in these fields. This webinar is going to be recorded, and the recording will be posted on the ASCCT website a few days from now. And your lines are muted. You can ask questions of the presenters using the Q&A feature at the toolbar at the bottom of your screen. Please put your questions in the Q&A so that we can find them, not the chat. There's going to be a Q&A after each speaker, so go ahead and get your questions in while the speaker is talking, and then we can address them. And I would encourage you to say hello in the chat and put in comments and chat with your fellow toxicologists. So I just wanted to briefly give you a preview of a couple of upcoming events. First of all, the ESTIV Congress is going to be in November in Barcelona. You can see the website there, and they have abstract submission open with a deadline of May 1st. So get your abstracts in and be part of this, what looks like a really great program. Also, the ASCCT is going to be having its annual meeting probably in October in the Research Triangle Park area in North Carolina. And actually the organizing committee is meeting tomorrow to begin planning the session topics and set the date. So stay tuned for more information on that meeting very soon. We also have a couple of upcoming webinars to keep an eye out for. On Thursday, the 14th of April, we're going to have a couple of presentations about new kinds of models for respiratory sensitization and methods to evaluate developmental neurotoxicity models. And that registration should be open pretty soon. It's not yet up on the website, but should be soon. You can always find information on upcoming webinars and recordings of all of the previous webinars on the ASCCT website. So our theme today is complex in vitro models, and we have three very nice presentations on this theme. First of all, Sabrina Madiedo-Podvršan is a PhD candidate at the Université de Technologie de Compiègne in the biomechanics and bioengineering laboratory. She's currently finalizing her thesis on the combination of tissue engineering with multi-organ chip technology. She's previously held a 2-year bioprinting research position with the L'Oreal team and is looking for opportunities to contribute to the field through a postdoctoral research position when she finishes her PhD. And Sabrina actually won the ESTIV Best Poster Award in recognition of an outstanding presentation of a scientific poster at the 2021 edition of the Joint Research Center Summer School on Non-Animal Approaches in Science. So congratulations, Sabrina. Our second speaker, Dr. Samuel Constant, is a co-founder and chief executive officer of Epithelix, which is a Swiss biotech company specializing in tissue engineering. Epithelix is a leader for in vitro assessment of drug efficacy and toxicity on human respiratory tract. Epithelix has developed unique 3D in vitro human airway tissues and testing services for studying pathologies like asthma, cystic fibrosis, COPD, and bacterial and viral infections. Samuel is in charge of global management and business development of the company and also deals with external collaborations with private and public research groups. And his team have won more than 18 prizes for their scientific and technological innovation and business development. And then last but not least, we'll have a presentation by Patrícia Zoio, who is a PhD candidate at the Institute of Chemical and biological technology in the Nova University of Lisbon. Patricia holds a master's degree in biomedical engineering with a focus on clinical engineering and medical instrumentation from the University of Lisbon, and a postgraduate degree in startup research from the Nova School of Business and Economics. Currently, she works on the design and fabrication of organ-on-chip platform with integrated sensors for the development of full-thickness human skin. And Patricia is also the winner of the ESTIV Best Poster Award in recognition of scientific excellence, innovation, and clarity of poster presentation at the 11th World Congress on Alternatives in Animal Use in the Life Science. So without further introduction, I will go ahead and give the floor to Sabrina Madiedo-Podvršan, and look forward to your presentation.
Thank you. So I'll be sharing my screen. Okay, can you see?
Yes, looks good.
Okay, perfect. Thank you for having me today. I'm very happy to be here to show you a bit of my work. As Christy said, I'm a final year PhD student in bioengineering at the University of Technologie of Compiègne. Today I'll be talking to you about my thesis, which is the development and the investigation of a new in vitro lung-liver co-culture model for the toxicological study of inhaled xenobiotics. This project is multidisciplinary and gathers both UTC and INERIS teams. In order to give you a bit of an overview of our work, I will begin my presentation by introducing to you the context in which our project arises, the biological, regulatory, and social sanitary framework of our study. Then I'll outline the objectives of our work focused on two pillars, experimental robustness of the developed device and its potential for practical issues. I'll then talk about the implementation of the project articulated in 4 experimental phases. And finally, I'll give you a better insight by briefly presenting a few results and our visions for the years to come. The respiratory system is exposed daily to a wide range of airborne xenobiotics. This exposure can be associated to adverse effects. Because toxicity occurs at various levels, in order to consider full and final potential of a substance, a multi-scaled assessment of associated toxicokinetics is necessary in order to obtain accurate prediction of risks. Local or systemic behavior can be tied to different types of biological responses, such as aggravation, stabilization, or protection of our body to exposure. Mechanisms involved in toxicity can often be complex. That's why animal models have traditionally been used as prime study models. For the time being, the current challenge in the complexification of alternative in vitro models to animal experimentation is based on the ability to connect different organs together, from the organ on a chip to human or body on a chip. So it's in this context that our project stemmed. We developed a new device that enables in vitro co-culture of a pulmonary barrier, which is the route of entry of inhaled xenobiotics, with a detoxifying organ such as the liver. This approach allows us to consider inter-organ crosstalk as possible modulators of toxicity, and therefore acknowledging toxicity at a more systemic level when it comes to evaluating associated risks and hazards of inhaled xenobiotics. Our project is also part of a social sanitary context in which we evolve daily, which is characterized by constantly evolving urbanization, globalization of our way of living, associated with the intensification of human activity and international exchanges. Both phenomena create, complexify, and multiply pollution sources, which has notable health impacts at a global scale, which propels the social sanitary need for respiratory toxicology. So our project is also incorporated in a wider context of reduction of animal use in science with the 3R, the European 3R rules. The development of reliable alternative methods has become a necessity. In vitro experimental models are good candidates because they're simpler and less expensive to implement than in vivo models. And they also give the possibility to work with human cells or tissue, which allows for better approximation of vivo physiological conditions and therefore improves translational vitro to clinical data. We're currently still in the development phase of our co-culture platform, which is why experimental robustness is at the heart of our project. Before being able to use this model for real case studies, we needed to, first of all, develop viable and functional tissues in an individual and joint culture setting, and highlight, differentiating, and characterize tissue-specific behavior from dynamic inter-organ crosstalk. Once monocultures and co-culture behaviors are comprehended, the lung-liver co-culture platform can be used to investigate common health concerns such as pesticide exposures. So this project is articulated in several experimental phases. The main goal of my thesis was to use a model substance to highlight passage and circulation of a xenobiotic through the device in order to show the relevance of both compartments to respond to xenobiotic stress. So the first phase was to characterize tissue-specific toxicological behavior of both lung and liver monocultures. To do so, viability, functionality, and metabolic activities of both compartments were separately assessed following exposure to a model substance. The second phase was prior to co-culture, preparing both monocultures to the joint culture setting. So that goes by choosing common media and other important parameters in order to best preserve optimal viability and functionality. Phase 3 is once these essential parameters are optimally configured to ensure proper culture conditions, both compartments were united in the co-culture platform and exposed to the same model substance than monocultures to assess any associated crosstalk behaviors. And lastly, the developed device can be used in a more physiological way by changing exposure modes to an aerosolized exposure and complexifying tissue by using primary or stem cell-based tissues. Both improvements to increase the accuracy of our model in order to empower its predictability when it comes to investigating controversial atmospheric pollutants such as pesticides. Due to time limitation, my presentation will focus on phase 1 and 3. A brief technical insight of our project. The lung compartment is reconstructed using Calu-3 bronchial cell line. Culture is carried out on semi-permeable transwell inserts. Hepatic tissue reconstruction is done using HepG2C3A cell line and takes place in microstructured PDMS biochips, which provide a dynamically stimulated environment. Pulmonary inserts and hepatic biochips are matured separately and before being serially connected in the co-culture box, which you see on the right. This specific culture device was developed in the UTC Biomechanics and Bioengineering Lab in 2004. This platform is a polycarbonate-based box able to accommodate 3 parallelized co-cultures. It's composed of a bottom part comprising wells for the 6-plated size transwell culture inserts and provides attachments for the biochips and associated media reservoirs. And the top part is for tubing. Both parts are joined and sealed together with a silicone seal and screws. In the specific context of our study, dynamic co-culture is maintained for 72 hours at a flow rate of 25 microliters per minute. So the present co-culture model was characterized using a well-documented hepatotoxicant called acetaminophen, also known as paracetamol, and for this presentation, I'll be calling it APAP. Exposure to the pulmonary barrier is not accurate, but allows to visibly observe passage and circulation of the xenobiotic through the device, as APAP interferes with hepatic viability and metabolic performances. We developed two kinds of lung liver models. First of all, the development model, which allowed us the technical setup, where the bronchial tissue is classically maintained in submerged culture during the whole period of the experiment. Chosen concentrations for this model rely on documented hepatotoxic thresholds. The APAP solutions are deposited on the pulmonary barrier at concentrations recalled as being local. When APAP passes the barrier to join dynamic circulation within the device, it's further diluted in the circulating media, totaling at 10 milliliters, hence decreasing the so-called systemic concentration to 1.5 and 3 millimolars. The second model is the physiological model, which better approximates a human vivo situation. Protocol includes an air-liquid interfaced differentiation stage and exposure mode. Exposure concentrations are lower as the locally deposited solutions are smaller, 200 microliters, to mimic a further physiological exposure mode. Local exposure concentrations are set at 0.5 and 1 millimolars, as it is the maximum solubilization threshold at this volume. So here are a few exposed samples of the collected global collected data for this model. In a monoculture setting, it allows us to outline a state of the lung liver compartments according to culture conditions. The bronchial and hepatic monocultures both respond to APAP exposure. Results provide evidence of a notable adverse effects on both tissues. Not only does the viability of the lung compartment on the upper left graph shows us tissue response to exposure concentrations as rates decrease with exposure, but also on the upper right part, functional transepithelial electrical resistance assay confirms that barrier function is impaired as measurements significantly drop. On the hepatic side, post-exposure cell counts highlight a significant decrease in live cells, and exposure was correlated to higher glucose consumption, which speaks for greater basal cell metabolic activities. Despite the apparent cytotoxicity imposed by the 72-hour exposures, both tissues remain live and functional. So as previously mentioned, adjustments such as choice of common culture media were dealt with prior to co-culture. This time comparing the previously presented data, monoculture data, to this time the co-culture data, which is here in with little dots. In a co-culture setting, so the condition in bold, APAP continues to display a cytotoxic impact on both compartments, bronchial viability through mitochondrial activity and hepatic viability through cell counts decrease significantly. We can also see here through the TR measurements how the bronchial barrier is altered as resistance drops following exposure. However, this effect tends to be attenuated in the co-culture condition. Trends remain the same from co-culture to monoculture, but difference in significance between non-exposed and exposed samples are diminished as statistical analysis of data highlighted. What's interesting is that this attenuation is accompanied by an increase in hepatic metabolism activity. So these biological responses tend to suggest that there is a lung-liver crosstalk associated with active and functional detoxification activity benefiting to both pulmonary and hepatic compartments within the lung-liver co-culture. This time, looking at the physiological model, Our goal with this model is to test its limits by exposing it to lower concentrations. At such low concentrations, we know that there will be no impact on tissue viability or functionality as expected. Nevertheless, interestingly, APAP metabolites were detected within the device, which attests of a passage and a circulation of the xenobiotic inside of the co-culture platform. So we do have an active metabolism. This study is still in progress to better quantify the sensibility of the device. But the good news is that this model does exhibit sensibility to low APAP exposure concentrations. So, so far, we have established co-culture is stable for over 72 hours, despite imposed stress. We are able to demonstrate in active and functional crosstalk within the device, and it seems to be sensitive to low doses of exposure, which is relevant for physiological exposures. This device can be used in practice with more physiological aerosolized exposure modes, and can be used to study in practical studies of, for instance, we were thinking of pesticides. So we hope to contribute to a new kind of toxicological approach, which would be valuable to build more knowledge towards better hazards and risks management regarding increasing global exposure to atmospheric pollution. Thank you for your attention, and I'd be happy to answer any questions.
Thank you, Sabrina. That was a really nice overview of your work. We don't have any questions so far in the chat, in the Q&A box. So maybe we could chat a little bit while we're waiting for any questions. I'm wondering if you have thought, it looks like you're looking to look into assessing pesticide risks. Another potential area that this might be really useful for is fragrances, sort of personal care product sectors that have airborne products. I wonder if you've thought about that at all.
We did. We'd love to extend the portfolio of tested substances, and actually the pesticide that we were thinking of is permethrin, which is a pesticide, which is an insecticide that's actually used at home. So anybody can really be exposed. So that's the type of exposure we're also interested in. The exposure that's, if I can say, discreet, that people can be exposed to without knowing it every day. So definitely the fragrances and that type of substances are interesting to investigate.
Great. Okay, we have a few questions coming in. So one of them is, how did you decide to choose APAP?
Thank you for the question. Very good question. We decided to use APAP because we have a strong, already we've been working with APAP in the lab for a few years, so we already had a strong base, and we knew that it had a significant impact on hepatic cells when cultured in biochip. And we chose it because it was a way for us to observe adverse effects at the level of the biochip within the co-culture platform. So that would allow us to know that APAP was able to cross the barrier and get to arrive to the biochip. So that's why we used this specific molecule.
Okay. There's a question about whether the lung culture was characterized in terms of biomarkers and whether it grows cilia.
So Cal-U3 for our culture, we did different types of microscopy. We weren't able to see cilia. We know that it produces mucus. We did immunostain tight junctions, so it does have a very good barrier function. So we have that. We know that we'd like to work with different type of pulmonary barriers. We also started investigating alveolar type tissues. So yeah, so it's a work in progress, but yeah.
Sorry, put myself on mute there. Someone asks about culturing Calu-3 cells for a longer period of time, saying that they've had some issues where they detach from the membrane after about a week.
Well, we have the opposite issue. I cannot detach them from the membrane. I've tried by treating them with trypsin, but the initial density in which I seed them, and I use a 11-day culture prior to co-culture, so adding 3 days is a total of 2 weeks, and the culture remains stable, no detachment whatsoever. So no issues for that. I haven't tried longer. It would be interesting, but for 2 weeks, we didn't have any issues.
Great. Another question about why you chose acetaminophen in the lung cell line.
Well, as I mentioned it before, it's because we've been working with it for a while, and we knew that it would have adverse effects on the biochip, so would allow us to see a passage in circulation.
Yeah, okay. And just a suggestion or a comment about using complex mixtures of pesticides in your scope. That presents a lot of challenges, I think, but it's more and more of interest for regulatory application. And I'll take one more question, and then it's wondering, there's someone wondering if you've thought about meeting regulatory requirements for pharma. It seems like you're thinking in that direction, but they're wondering if you're thinking to validate the system and then use it for preclinical inhaled drug.
It would definitely be the optimal final goal, but we are still in the very early stages of development. So for now, it's not the priority, but for sure, it would have to be tackled at some point.
Yeah, okay. Okay, there's some other really good questions, and maybe you can address them while the next presenter is talking.
Okay.
So
can I answer by on the chat directly, maybe?
Yeah.
Okay, perfect.
Yeah.
Thank you very much.
Okay. Great. Okay, so next we'll go to Dr. Samuel Constant. Sam, if you want to share your screen, the floor is yours.
Yes, with pleasure. So first of all, hello everybody. And I'd like to warmly thank Christy and all the team also for, let's say, providing me this opportunity to talk with you about our activity at Epithelix. One part of our activity is dealing with toxicity assessment of inhaled compound in general, and the other part is more about efficacy, and we have a different aspect on research on pulmonary diseases. And one of these subject is really focusing on antivirals development, and this is one of this subject that I would like to present to you today, is how we could leverage 3D human airway epithelial model to study the well-known now SARS-CoV-2 pathogenesis, and to discover a new antiviral which could be potent to solve these issues. But before starting into this subject, I'd like to discuss a bit on facts on COVID-19. Well, we all know that the first case were discovered in Wuhan in December 2019. And it's very interesting to see how the scientific community has reacted fast. And indeed, because already in January 2020, the genome was revealed, and we have access to the first PCR diagnostic, which was published. In March 2020, meaning that almost 3 months after, we have already the first antibody test, and in May, the first antiviral authorizations given for emergency use of one antiviral, which was remdesivir, given by FDA. So it was really incredible to see that in a short time frame. So, but what is the situation today, as of today? At least in EU, we have 5 vaccines which are authorized by EMEA, which is really great. We would not have bet that, you know, such a short development, very fast development, so to have access to the wide community of these vaccines. And also 2 new antivirals have been authorized. One is Paxlovid from Pfizer, which had authorization from both FDA and EMEA, and the second is Molnupiravir from Merck, today for the FDA, and waiting for the European authorizations. But also the numbers of research articles was really incredible. It's really impressive to see how scientists really are focused on this research, because according to the WHO database, there is more than 400,000 research articles on COVID-19 since then. So it's really a huge amount of data. But the situation is not is really dramatic, because there is more than 457 million cases which have been reported, and it seems to be underestimated. And among these cases, as you could see here, 80% of the cases are really mild symptoms, 15% are severe symptoms, and 5% extremely severe symptoms. And it gives rise that, unfortunately, it's a really deadly situation, because there was more than 6 million as of today's deaths worldwide, and give rise to 1.3% of death rate. It's not just a simple flu. So now, looking at this fact, the reason of this is this virus, which is SARS-CoV-2. So SARS-CoV-2 is an RNA virus with the variant diameters of between 50 to 200 nanometers, which is formed by proteins. So S, E, and M proteins are forming the viral envelope, and the N proteins here inside hold the RNA genome. So the most known protein is obviously the spike S protein, because this is the one on the surface of the virus which allows the virus to attach to and also to fuse to the membrane of the host cells. So how does it fuse and attach? Well, first we need to interact, like all the viruses, with a cell surface receptor, specific receptor. And the one for SARS-CoV-2 is ACE2, angiotensin converting enzyme 2, which is present here. But so once you have attachment first of the virus at the surface of the membrane, it cannot fuse by itself. It needs also other helpers, molecules present on the surface of the cell, such as a transmembrane protease serine 2, an enzyme here, which has the responsibility to cleave the spike protein, and then you have a fusion of the virus inside the cells. So once you have that, here you have the virus which could enter into the host cells. The most straightforward cells to have these replication cycles are airway epithelial cells here. So that when you breathe, you have these interactions, and then you have the virus which could replicate and to form new viral proteins, and then which could mature into new variants, which could be released, vectorial release apically, and then when you spread out this new mature virus, it could infect again neighboring cells. And for this reason, this is really an exponential growth of the virus, because you could produce more and more from neighboring infected cells. So what's the situation classically? Well, in a normal situations, the airway epithelial cell is able to respond to a danger, to respond to the virus by secreting a signal such as interleukin. So there is a panel IL-6, IP10, for example. And there, the reason of this activation is that they could recruit effector cells, such as T cells, monocytes, and macrophages. And they are here in order to clear the virus, which is here, and also to phagocyte, such as the alveolar macrophages, which we see here, to phagocyte the apoptotic cells. So in an healthy immune response, the infected cells are rapidly clear, the virus are inactivated by neutralizing antibody, for example, and there is at the end, as a consequence, a minimal inflammation and lung damage. But in a abnormal, a dysfunctional immune response, unfortunately, you could have an excessive infiltration of monocytes, macrophage or T cells, for example, and then induction of systemic cytokine storm, and in the worst case, pulmonary edema and a widespread inflammation and multi-organ damage. So, now coming to in vitro. So at Epithelix, we have been working for many years now in order to model different part of the respiratory tract, such as the nasal epithelium, tracheal epithelium, and bronchi epithelium, with a model which is called mucilaire, which is made of basal cells, ciliated cells, goblet cells, present cilia, fully ciliated on top, and mucus, and sculpture in inserts. So we have been also interested in generation of small airway epithelium with a model which is called small air, including a new type of cells here, which is cuboidal club cells. And even more recently, we have been working also on the alveolar models, which is quite different because it be a pneumocyte of type 1 and type 2 and produce surfactants, but also in insert culture support. So all of these models that we produce as mass and standardized at Epithelix are made of primary human cells taken from surgical operations. We use very low passage cells here, and in the process we amplify them, but only once. That here we could freeze the cells and seed them later on when thawed onto this insert support, and we let the suture cells to the liquid interface, and they progressively differentiate to the pseudo-stratified epithelium, and this process takes around 6 weeks. Well, we could produce nasal, tracheal, bronchial, small airway versions here, but I will concentrate here on one which is widely used for virology, which is mucilaire nasal, but not from single cells, not from disease patients, because using primary cells you could have disease patients such as asthma, CF, and others, but here only with normal pool of donor of normals, which could minimize inter-individual variability and a large batch possibility of productions. So this mucilaire, especially the mucilaire pool, has been extensively used in the past prior to SARS-CoV-2 for infection models such as rhinoviruses, infection, influenza research, respiratory enteroviruses, RSV, metapneumoviruses. But for coronaviruses, SARS-CoV-2 here is the main interest of today's talk. So first, I discussed about the ECE2. So we needed first to prove that this receptor was present, and it was the case here in the nasal versions of the mucilaire, bronchial version, but also the small airway, where we could find here a large expression of ACE2. Working on isolated strain collected from hospital, so we were able here to infect with living viruses, see the replication of this virus with time, so they could be used as a reservoir to produce newly generated SARS-CoV-2, and also to study their pathogenesis. Very interestingly, at the same time, you could monitor the replications. So here you wash off the apical surface and then you quantify by RT-qPCR the amount of replicated viruses, but also at the same time, you could also look at the influence of the barrier functions monitoring transepithelial resistance, which is a very good index of tissue integrity and epithelium damage here. So look at cell tropism. So it is possible to on this pseudostratified epithelium to do cross section and electron microscopy and mainly to make it short, SARS-CoV-2 mainly infect on the upper respiratory tract, ciliated and goblet cells, but to a really fewer extent basal progenitor cells. Interestingly, and this was published in Nature, that the epithelium, pseudostratified epithelium, is really fully ciliated, as you could see here. But upon infection with SARS-CoV-2, you have a disruption of the cilia surface here, and it was transmitted into function. One of the main functions of the upper respiratory tract is to trap airborne pollutants inside the mucus and to remove this trapped pollutant thanks to the impulsion of the cilia beating with mucociliary clearance. And you can quantify this mucociliary clearance here, and this is a classical distribution which could be found, but in presence of SARS-CoV-2, there is no more movement, meaning that SARS-CoV-2 for long-term effect, you have this strong impairment of these mucociliary clearance functions. So the airway epithelium is not only a physical barrier, but it's also a highly potent immunoregulator. And so innate immune transcription, as in Nature, was studied here in presence of SARS-CoV-2, and several genes were highly upregulated. And one of those were type 1 and type 3 interferons, and also genes associated with NF-kappa B and TNF-alpha, which could be used as a good signal to see effect of antivirals also. So looking at antivirals, indeed, today, what are the current strategy to stop SARS-CoV-2 infections? Well, you can test monoclonal antibodies or convalescent plasma, for example, to act directly on the virus, but also look on the receptor. For example, if you develop a series of receptor inhibitor, for example, one of the most known is camostat mesylate. You could interact directly, and that reduce the amount of virus which could be found inside the host. But there are also other type of mechanisms which could be studied, such as inhibition of protease activity, inhibition of RNA polymerase, and others. And all of this could be studied in this already simple in vitro models. So the airway epithelium like this in insert is really tight. And the advantage is that you could compare different delivery. So you could test a basal exposure of your antiviral in order to mimic systemic delivery, meaning that if you imagine that your new antiviral would be administered through IV or through pill, then it would be an ideal exposure scenario. Or you could apply your compound apically or formulation apically to mimic either a nasal spray or a nebulizing chamber, for example. And the form of the antiviral could be in different ways. So you could test liquids, solids, gas, and nanoparticles if needed. Now, what type of endpoints are relevant for monitoring antiviral in SARS-CoV-2? Well, the first is viral copy genome number, because you apply first the virus, and then you remove the first virus that you have applied, and you collect at different time points here a newly generated virus which was replicated at different points. Okay, and then this could be easily quantified by RT-qPCR methods. At the very same time, it's important to see the effect on tissue integrity, because if you kill the cells will less replicate virus. So it's very important to distinguish between toxicity and antiviral activity. You could quantify infectious titers, gene expression analysis, also scanning electron microscopy, and a secretion of soluble factors such as cytokines, chemokines, and metalloproteinase. So many different antiviral approaches have been tested within the last past 2 years. So I've been working on more than around 500 compounds and formulations. But I will list here some very active compounds here that we tasted, and you could select the dose which could be interesting here in terms of activity. So to make really short, interestingly here, molnupiravir, which is here, and Paxlovid, this is one of the analog of Paxlovid, which is here, were found to be really highly active in this model. But we can even do better than monotherapies. And part of our research activity was also to try to enhance the activity of these monotherapies. So we use as an anchor molnupiravir, which is the one produced by Merck, and try to see if combination therapies could enhance this antiviral activity. And it tends to be the case because not all the dual mechanism work, but some of them yes. And for example, there are reported case for which, for example, when you add on top of molnupiravir different other compounds such as ivermectin, camostat, brequinol, then you could have an enhanced activity, which is let's say increasing by 1 or 2 log increase of the antiviral activity, which is really good and promising. So this is where we are for this combination therapy for the moment. Now discussing a bit about predictivity, because all the models need to tackle these issues, all in vitro models especially. How do we translate to existing in vivo data? So which is not always an easy task, and we have to compare with also a gold standard also and the gold standard in virology is Vero E6 cells, so green monkey cells, and in one of the cases, which was with hydroxychloroquine, which has been found to be effective on Vero E6 cells, also at high concentration, with a mucilage system, it was not found to be active, at least at subtoxic doses, because at toxic doses, obviously, it was the case. And there was studies, in vivo studies on cynomolgus macaque shown that it was either not effective in these macaques, and we all know that also in human clinical trials it was not the case also. So indeed, so there was a very good correlation between the mucilage data and the in vivo data, but a bad correlation from more simple models such as this Vero E6 cells. So it shows show that working with physiologically relevant and untransformed models, so you may obtain here very predictive data, at least for this purpose. So as a conclusion, I hope I have convince you that air liquid interface 3D human airway models are useful tools to study SARS-CoV-2 pathogenesis. Well, you could use them to isolate, characterize, and amplify circulating strains. So this is a bioreactor, if you want. But they are very useful for identification of transcriptional and secreted innate immune signature in order to better understand the mechanism and the mode of action of the pathogens, and for the pathway identifications. And very importantly, because we want to cure, we need to cure. So there are a very good screening platform to test antivirals, either for the repositioning of marketed drug, which was the initial let's say, idea and channel of researchers, but also to evaluate novel therapies and combinations, which could be delivered either systematically or through aerosol therapy. And the advantage is that you could locally have access to information of the local toxicity simultaneously with efficacy at the very same time. So I'd like to thank all my collaborators, great team from the French labs, the Swiss labs, collaborator also from VIRPAS, DSL3 in Lyon, University of Geneva, Spitz Laboratories, and funding from the Gates Foundations. If you are interested in knowing more new updates on in vitro long model approach, I welcome you to this the live meeting, which will be organized in Nice the 13th and 14th of June. Please be there if you want to have a new update. And I would like to warmly thank you for your attention.
Thank you, Sam. That was really wonderful. There's just so much work that Epithelix is doing to address COVID. So it's really nice to see. We have a lot of questions for you so far, but I also wanted to put a plug in for your conference. I think we attended the one a couple of years ago before the pandemic. It's a really good conference, and I've got my abstract drafted, so hopefully everyone else will as well. Okay, so let's see. There's a question about a brief question about using airway models for respiratory sensitization. Wondering if you have any comments about that.
Well, there are very good initiative today. There is one model which is under review by a group of LIST, Arno Gutleb lab, with a tetraculture incorporating all cell line based tetraculture with A509, THP1, and others. The set of data being not huge today in terms of respiratory sensitizer testing, but they are providing results for the moment. So this is a good news. Yes.
Great. A question about exposure to gases, and they're wondering with the model, how do you make sure the gas stays in contact with the cell culture?
Very good questions, and for this you need to have very good devices. So there are some commercially available options. So the insert systems, the advantage of the insert system, it fits in different exposure modules. So one of those is, for example, VitroCell exposure devices where you have let's say mixing chambers with gases and inert air that you could dilute as a specific dose and go directly on top of the surface of the epithelium. And for it's generally you have some pulses for several minutes to even more, and then you go back to the incubator and you could do repeated exposure like this.
Thank you. Someone else is wondering, the slide that you showed where there was no correlation between the Vero cells and the mucil air model, do you know why, or do you have any hypotheses why that might be the case?
Yes, one of the, in vivo, we are always fighting against viruses in general, if you think about that. And not all the viruses are infecting us. And the reason is that you have an innate immune response, which is active at the epithelial level in vivo. And we are secreting defense molecules such as interferon, which is one of the most active for viruses, and the constitutive levels of interferons on the airway epithelium is really good, as you could have in human, and this is not the case for the Vero E6. So it could explain why you ended up with a positive effect and negative on some angles.
Yeah. Okay. There's a question about mentioning that COVID therapies are often administered orally or via IV, and just wondering about, you know, I know you mentioned potentially inhaled therapies, but just wondered if you have some comments about that interplay between the different exposure scenarios.
Yes, we had a lot of cases where we found that some compounds are more active in inhaled therapy than others in systemic therapy. There are several reasons. One is intracellular trafficking, because you have active pump, and some compound could pass easier inside the epithelium, being from the systemic or from the apical. It's really depending on the compounds, really compound specific. So this is one of the examples. Other could be if you inhale a compound, generally you could act directly on the site, so you could minimize the amount of drugs which could be applied in you. And this is one of the examples, as Sabrina has introduced before. The liver toxicity is important, and sometimes it's a drawback for a drug, you know, could fail because of this very high liver toxicity, and because if you had a high amount of drug in the systemic, so you may generate a metabolite which is more toxic, and it's a way here to minimize this hepatocyte-induced toxicity by delivering directly a lower amount directly on site.
Okay. All right, so one last question and then we'll move on to the next talk. It says that Calu-3 cell lines are widely used for SARS-CoV-2, and wondering if you could explain the added value of 3D models like your models to, you know, above those types of models.
Well, there is another Nature paper where they use Calu-3. Calu-3, indeed, they are used, but if you want to validate, you validate them onto 3D primary human airway epithelium. So I recommend one of the very nice Nature papers that I could link here on the post. This is pretty much clear that you would have you could reduce the number of targets using this epithelium. One of the reasons is what I explained before. The defense mechanism is not the same. The internalization is not the same. Ciliogenesis, and Sabrina has also mentioned that. So you infect ciliated cells. If you have no cilia, there is a bit of drawback in the analysis. A bit of to be as close as possible to the physiological system. It's very important, I think, to use a primary cell-based approach. So a lot of pre-screen on Calu-3, And then you confirm on more predictive models such as the BCR.
Right.
This is the way that industry is doing now, and yeah.
Tiered testing strategy.
Here's testing strategy.
Yeah. Okay, great. So thank you so much for your presentation. Thanks for answering all those questions. Really great discussion. Before we turn it over to our next presenter, I just wanted to mention in the chat early on, Sue Leary from Alternatives Research and Development Foundation is mentioning they have their open grant deadline of April 15th. So they provide grants for in vitro and in silico models. So we'll put that link again in the chat in case you joined later, but I just wanted to highlight that funding opportunity. Okay, without further ado, we'll move on to Patricia Zoio. Would you like to share your screen, Patricia?
Thank you. Can you hear me okay?
Yeah, sounds great.
Okay. Okay. Can you see my, can you see it?
Yes, looks good.
Okay.
Please go ahead.
And so thank you so much for the opportunity. So today I'm going to present my work entitled Full Thickness Skin on a Chip Model for In Vitro Drug Testing. This work was performed at ITKB Nova in Lisbon in the context of the Advanced Integrated Microsystems PhD project. So the main motivation for this work is the increasing demand for physiologically relevant in vitro skin models. And this is due to multiple factors. We have the ethical regulations, as you know, which encourage replacement of animal testing according to 3R principles. At the same time, you have the increasing prevalence of skin diseases. So we have chronic problems such as psoriasis and eczema. And at the same time, we have the malignant diseases such as melanoma, which unfortunately are frequently fatal. And this increasing prevalence of skin diseases is fueling the skin targeted drug delivery market, which includes topical, dermal, and transdermal approaches. To test these pharmaceuticals, conventional in vitro skin models are typically used. So we have 2D and 3D cell cultures approaches. In 2D cell culture, typically it is monolayers of keratinocytes or fibroblasts, and these systems really lack the 3D complexity of the native skin, but have the advantage of high throughput and reproducibility. Then you have the conventional 3D skin models, the epidermal models or full thickness, which means they include the epidermis and also the dermal compartment. And they, in fact, increase the complexity of these models, but really lack the reproducibility approach. Furthermore, these models are known to have weaker barriers than the native skin, and they lack mechanical stability, mainly because they typically use animal-derived materials such as collagen, which tend to contract over time. Organ-on-a-chip technology promises to increase the predictive power of in vitro models by increasing its physiological relevance. This technology combines cell biology, engineering, and biomaterial technology to simulate the microenvironment of tissues and organs by mimicking the complexity of the tissue's interfaces and also the mechanical stimulation that cells sense in vivo. And this really has the potential to increase the physiological relevance of these models while maintaining some reproducibility, some throughput. So the goals of this project that I'm going to share were the development of physiologically relevant full thickness skin models. And there were two major goals. First, I wanted to develop a full thickness skin model under conventional static approaches. However, these models should have enhanced differentiation, enhanced mechanical stability, and should not use animal-derived collagen, animal-derived hydrogels. And I wanted to study its applicability for drug testing. Then I want to adapt this model for production of skin under static conditions to the development of a full thickness skin on a chip. I first designed and fabricated an organ on a chip to establish dynamic perfusion on the tissues, and I integrated sensors for real-time evaluation of the skin barrier. So starting with the goal of developing a skin model with enhanced mechanical stability. This was achieved by combining a porous polystyrene scaffold with the concept of a fibroblast-derived matrix. In this concept, fibroblasts are seeded into the scaffold and they are stimulated to synthesize their own human extracellular matrix, thereby avoiding the use of animal-derived collagens. And this way, we can achieve a full human tissue. This method was used to design, to develop and optimize a protocol to generate a full thickness skin model. So the first stage is the development of the dermal compartment using the technique of the fibroblast-derived matrix. Then we seed melanocytes on top of the dermal compartment to achieve skin pigmentation, followed by epidermal formation, first under submerged conditions, and then at the air-liquid interface until a stratified epidermis is produced on top of mature dermal compartment. And the first thing that we did was to optimize the dermis culture time to achieve a mature dermal compartment, which would be capable of supporting an epidermis on top, while allowing keratinocyte infiltration into the dermal compartment. So you can see here the dermal extracellular matrix proteins over time, and as expected using this approach, the fibroblast-derived matrix approach, there is an increased expression of collagens and fibronectin with culture time. We also saw that these dermal maturation times affect epidermal development, so keratinocytes were seeded on top of dermal equivalents cultured for different times. And you can see when keratinocytes are seeded in 3 days dermal equivalents, there is a tendency of the keratinocytes to penetrate the dermal compartment, resulting in the epidermal disorganization. From 6 to 18 days, it is possible to obtain a differentiated and organized epidermis without signs of infiltration. So we chose this condition, the 6-day dermis, in the following experiments. Overall, using this technique, it is possible to obtain a good quality skin with a stratified epithelium structure in the layers that you can see in the native skin, in the stratum basal, spinose, granular, and corneal layers, also known as stratum corneum. In parallel, we developed also an epidermal compartment, which was also evaluated in parallel. We then kept these models at the air-liquid interface for up to 50 days, and we monitored the structure over time. We concluded that these models have extended lifespans compared to many conventional models reported in the literature. So you can see here that at 30 days, there is a tendency for a reduction in epidermal thickness. However, there is a preserved architecture. At 50 days, the models tend to remain stable, maintaining the same thickness and epidermal layers. And very importantly, there are no signs of damage on the dermal compartment. There are no signs of contraction or damage. On the contrary, it continues to synthesize fibroblast-derived matrix. We also want to develop a pigmented version of this model, and it was achieved with active melanocytes at the dermo-epidermal junction. You can clearly see macroscopically the pigmentation of the model compared with the model without the melanocytes. And we also used markers to identify these cells in the histological layers. This part has been published in the tissue engineering, and the main conclusions of this component of the static model is that it is possible to produce a skin model with high mechanical stability, which does not use animal-derived hydrogel, such as animal-derived collagen. We achieved dermis formation in only 1 week, whereas other approaches that use fibroblast-derived matrix need 4 weeks. This model has an extended lifespan compared to conventional models, and it is possible to achieve a pigmented version of the model. this month, it will also be possible to see a continuation of this analysis, where you also perform drug testing on the model, and you could check it out this month on the Alternatives to Animal Experimentation journal. Okay, then we wanted to combine this concept of producing a full thickness skin model based on a fibroblast-derived matrix with organ-on-a-chip technology. For this, we designed and fabricated a skin-on-a-chip device that was capable to ensure stability during culture of the skin, while providing a modular, flexible geometry compatible with different types of studies. So you can see here the chip. It has three main layers. You have the central cell culture chamber fabricated from PDMS and the polystyrene scaffold where the cells grow. And then we have two chambers, the apical and the basal chamber, fabricated from polycarbonate to achieve a double perfusion system, which allows continuous supply of nutrients and removal of metabolic waste products. And this organ-on-a-chip device has been fabricated using CNC micromachining and laser cutting in combination. And this is what the assembled skin-on-a-chip device looks like, with blue dye showing the fluidic compartments. And you see here the PDMS cell culture layer where the cells grow. You can also see two skin-on-a-chip devices placed in fabricated dedicated supports. The protocol used to develop a skin-on-a-chip model is very similar to the one that I described to you before. However, here we establish dynamic perfusion inside the chip. So we see the fibroblasts into the scaffold that is integrated into the device, and we establish double-sided perfusion during some days to achieve the position of the extracellular matrix into the scaffold matrix. Then we have the step of epidermis formation inside the chip, first under submerged conditions with double-sided perfusion, and then at the air-liquid interface, meaning we pump air at the apical surface and we pump the medium at the basal surface until a differentiated full-thickness skin is produced inside the platform. And this is what the setup looks like. You can see here the reservoir with the cell medium and the perfusate and the peristaltic pump. And we achieve dynamic perfusion by connecting the chip's inlet ports to the peristaltic pump. And you see here 4 skin-on-a-chip devices being cultured. Our first main conclusion was that dynamic perfusion inside the chip stimulates the production of endogenous fibroblast-derived matrix. So you can see here that compared to the static culture, the dermis produced using dynamic flow results in decreased expression of both collagen type 1 and fibronectin. And this is for a dermal compartment cultured for 6 days. But this can also be seen for a full thickness skin model cultured for approximately 20 days. So you have here the skin-on-a-chip model, the static model, and increased expression of both collagen type 1 and also fibronectin. From an histological perspective, both models produced stratified and differentiated epidermis after approximately 11 days at the air-liquid interface. There are no significant differences regarding the thickness of the corneal layers. However, we see major differences in the thickness of the viable epidermis. You see here the skin-on-a-chip model is thicker than the full thickness skin model produced under static conditions. You have to keep in mind that here we used much higher cell passages than the first models using conventional approaches. So the model tends to have a decreased thickness in general. Also, the skin-on-a-chip model shows an increased expression of both involucrin and filaggrin, which is a skin barrier protein and differentiated marker. So this points to an enhanced differentiation of the skin-on-a-chip model compared to the static approach. This was confirmed by performing permeation studies directly on sheep. And you see in the graph that skin on a sheep models present significantly lower cumulative amounts of a fluorescent molecule compared to the static models, as well as lower permeability coefficients. So we can conclude that organ on a sheep technology produces skin with increased barrier properties. Then we wanted to integrate sensors into the device for real-time evaluation of the transepithelial electrical resistance measurements during tissue formation. So you can see here the electrodes concentrically installed into the device. And this is a very interesting technique. It can be used as a non-destructive measure of the skin barrier by reflecting its, by measuring the overall barrier to ions. And we use this technique to evaluate the stage of skin differentiation. And you see here that during a culture for approximately 2 weeks, there is in fact an increase in these transepithelial electrical resistance values, which reflects the progressive accumulation of the stratum corneum and also the gradual formation of tie junctions. And the obtained values are consistent with the barrier function integrity. We also wanted to study the potential of these methods of measuring the transepithelial electrical resistance measurement for performing skin toxicity testing. So we performed these measurements while exposing the skin to a benchmark irritant, in this case SDS, for approximately 3 hours. And you can see a decrease in tear values, which points to a disruption in the barrier function. This disruption in the barrier function was confirmed by performing both histological analysis and by studying the penetration of lucifer yellow dye in different time points. So, and this confirmed the impacts of the irritant on the skin barrier. So we concluded that drug testing can in fact be performed directly on the developed skin on a sheep platform with key diagnostic parameters, in this case, the skin barrier function, being monitored in real time. And this has been published in these 2 journals. Basically, we show that it's possible to produce a platform with a modular architecture that can integrate a thick porous scaffold. We show that dynamic flow during dermis on a sheep step can increase the position of extracellular matrix, in particular collagen type 1 and fibronectin. We produced a well-differentiated and organized epidermis on sheep that has an increased thickness and enhanced barrier function compared to the static models. And we show that permeation assays can be performed on sheep, avoiding the use of France cells and other devices. And we also integrated transepithelial electrical resistance sensors to monitor the tissue development. And we also show that this analysis can be used to perform skin toxicity assays on sheep. So overall, I think this shows the potential of the skin on a sheep platforms to increase the physiological relevance of skin models. In particular, this design, this approach could be compatible with the long-term formation of skin to mimic complex skin diseases and also to evaluate the safety and efficacy of novel drugs and technologies. And this design could also potentially be adapted to other organs, in particular barrier organs, such as the lung and the gut. So we really believe that it has some potential. I want to thank the Biomolecular Diagnostic Laboratory from ITQB, in particular my advisor, Dr. Bella Oliva. I also want to thank my PhD project, Advanced Integrated Microsystems, Dr. Joana Marto, FCT for the funding. And I want to thank you for being present. And if you want to contact me, you have here my email. And if you have questions, please just ask. Thank you.
Well, thank you so much, Patricia. That was a lovely talk. And I love the pictures that you have kind of showing what it looks like in the lab, right?
Thank you.
Nice. So we have a couple of questions for you already. One of them is, have you characterized the phase 1 and phase 2 metabolic capability of your model?
No. You have to see that this is the beginning. It's a preliminary model. Now it will be available some detailed characterization of the barrier properties using following testing guidelines from OECD. But this is as far we didn't characterize much more than this. This is just the beginning.
Yes, good. There's a question about tier measurements with your model.
Mm-hmm.
The full thickness skin matures in an air liquid interface.
Mm-hmm.
Yes, and is it submerged for the tier measurements?
Yes. Okay. During the tier measurements, you really have to pump liquid medium at the apical surface, so you can have the current passing through the. So at that stage, we pump medium at the air liquid interface.
Okay.
At the moment, you do that manually. In the future, we should have a different approach that to do that. We'll do that without pumping manually the liquid. Yeah.
I think they're wondering if you can, so remove the liquid and continue to mature the model after you do those tier measurements.
The model is matured. The model needs to be matured at the air liquid interface. That's the condition to produce a differentiated skin. But you can perfectly you can measure tier in seconds, in minutes. So we just pump liquid at different time points, and
Okay.
then we remove it, so it continues at the air liquid interface. Yeah.
Great. Okay, that's clear. Thank you. Is the skin model, or have you tried yet, or you're planning to try to assess the dermal penetration with the model?
Dermal penetration of different
drugs.
Different drugs. Yes, we did some preliminary studies, just very preliminary with the fluorescent. Do I have it? With some fluorescent molecules.
Mm-hmm.
So we just we have a module to test some drugs. I didn't show it, but the chip has different modules, so you can. We have a different module, so you can put a drug. Can easily pump a drug at the apical surface. So this is a penetration model. So we have your fluorescent molecule at the apical surface, and then we, at different time points, measure the amount that's penetrated inside the skin on a sheep. But this is just a preliminary, so it's just a fluorescent molecule. I did penetration, I did permeation assays, using the conventional model using France cells. Mm-hmm. So we did this for hydrocortisone. We did this for many molecules.
Okay.
And compare it with the Stratem, which is a membrane, a commercial membrane for permeation assays. And they have a very comparative behavior. So it will be available on a future journal in the Alternatives to Animal Experimentation. So,
okay.
Yeah.
Yeah, that's great. I know that would be very useful.
Yeah.
Okay, another question is wondering about the costs between the two models. So the first one and then the one within the organ-on-chip technology.
I would say that the organ-on-a-chip can be less expensive because the area is much, it has a smaller area, and you can better control the reagents, the medium, it's also less. And the chip itself, this design in particular is very low cost. It's made from cheap materials, polycarbonate plastic, basically. So in this design, you just have to, depending on the lab, you maybe need to invest on the peristaltic pumps. Well, that's maybe you need specialized equipment. But if you have that specialized equipment, I would say that it's not more expensive. I don't know. I cannot say at the moment the prices. but I would say that it's at least comparable, probably less expensive.
Yeah, that's very interesting. Okay. Another question is, what technique did you use to manufacture the porous scaffold?
We did not manufacture the porous scaffold. It is a commercial available. So we chose it based on these characteristics, the pore size and the thickness, and it's called the Epithelix. So it is available for many applications. You can use it for wherever.
Okay. And there's a question about whether you have, some of this work has been published, but you shared a few publications in your slides. So maybe what we can do is when we post the recording of the webinar on the ASCCT website, we can add links to some of the presentations.
Thank you. It will be a pleasure. Thank you.
Okay, and there's a last question about a different endpoint, skin sensitization.
We did not progress to perform skin sensitization, but it's very interesting, and we are. It is on the list of things to test. We performed skin irritation assays using the conventional. So we are trying to test the 20 the drugs, 20 molecules suggested by the testing guidelines. And we already did some. So we are trying to do the 20, and then we will progress to other types of analysis, such as skin sensitization. But at the moment, only skin irritation and some permeation assays.
Okay. Yeah, I think the skin sensitization, to capture all the key events, you would need to include dendritic cells and cells. So.
Yeah, at the moment, it's on the list.
Yeah. Well, thanks again for your presentation. Congratulations on your award and on your work. It's really, really nice amount of work. I'm going to go ahead and close the webinar for today. I wanted to thank all of you for listening. I wanted to thank our three presenters, Sabrina and Sam and Patricia, for sharing your work today. I also want to thank Phoebe Woodruff. She is behind the scenes, but she has been helping to make sure the whole webinar goes smoothly and we have everything we need. So thank you, Phoebe, for doing that. And I wish you all healthy and safe March, and we'll see you next month. Thank you all very much.